Presenter is Noah Earland from Southern Methodist University.
Okay. Good morning, everyone. My name is Noah Earland, and during the course of last summer, I had the honor and privilege to conduct research in the Dr. Dale Dorscheid lab under the watchful supervision of Dr. Sam Wadsworth on a project entitled Characterizing a Three-Dimensional Bioengineered Model of Chronic Airway Fibrosis. So to begin, pulmonary fibrosis is a degenerative disease that affects the human airway tissue, progressively stiffening it. And there is no effective cure. In fact, the only treatment option available for patients who have pulmonary fibrosis is lung transplantation. And typically, patients that do not receive a transplanted lung will die within 5 years of the onset of their disease. Now, pulmonary fibrosis begins with epithelial microinjury, which affects all types of cells along the human airway, and this is especially seen through the beginning of the loss of cilia on these epithelial cells. And then the next stages are intense matrix remodeling and fibrotic pathway progression. Now, pulmonary fibrosis is especially characterized by the appearance of the so-called myofibroblast phenotype, which is essentially like a fibroblast, except it has contractile abilities that are able to pull on organized collagen fibers and progressively stiffen the airway tissue. Now, to this disease, like most diseases, we used animal models in the past to study pulmonary fibrosis, and that's in accordance with something called the Croft principle. Well, essentially the problem, especially in pulmonary fibrosis, but in a lot of other airway diseases, is, and I mean according to this quote on the board from Alan Olaf, who is the former executive director for cancer research, that these predictive animal models aren't predictive really at all. And they've been kind of hampering drug discovery. And that's actually the percentage of all basic science papers in leading journals that used animal models to come up with a new class of drugs, and it's clearly very small. So when it comes to specifically pulmonary fibrosis, the main model you use is a bleomycin murine model. And the bleomycin is a poison used to incite injury on the mouse airway, and that's how you'd study the disease. However, when you look at the genomic responses to the bleomycin model, you see poor translatability to the human condition. And I mean, when you actually compare the tissue types, it's really no surprise, starting with the fact that the human bronchial epithelium has a pseudostratified ciliated cell type with multiple cellular layers, notably the basal, parabasal, and apical layers, whereas the bronchial mouse epithelium only has a single cell line of a simple cuboidal epithelium. So they're very, very distinctly different tissues, and that's why you don't really make too many gains using the bleomycin murine model. So to this deficit, a group called BRONC, which is an academic group interested in researching bioairway construction, 3D bioairway construction, and it's actually headed by Dr. Sam Wadsworth, who was my supervisor in the lab. It's his own biotech startup. BRONC offers its own human airway 3D model. And so the full name is the 3D Airway Air-Liquid Interface Co-Culture. And it essentially begins with electrospun gelatin, and you see two different primary cell types on it. First, you seed your primary human airway fibroblasts, followed by your primary human epithelial cells. So, barrier function was a way of standardizing our normal 3D models before we introduced any sort of treatment to them. And barrier function was measured using something called transmembrane electrical resistance. And like any type of electrical resistance, it's inversely related to area, so that would be multiplied by centimeters squared. And in culture, you get your 200 ohms times centimeters squared benchmark, and that was seen as kind of the standard for a quote-unquote normal or untreated 3D model. And then at the bottom, I'd like to note the surprising similarities between an actual human airway wall and our own 3D model. Note the formation of the parabasal, basal, and apical cell types for the epithelium, and also the fibroblasts located directly beneath. So then in our 3D model, we also see this gelatin region, which the fibroblasts were free to migrate into and remodel as they pleased. So our lab hypothesized that by treating the airway models with profibrotic growth factors like TGF-beta will trigger the expression of fibrosis-related extracellular matrix proteins. Our specific aims went as follows. So this hypothesis was actually determined before I got to the lab, but I carried out all three of these specific aims. So first I had to optimize antibody concentrations for immunohistochemistry. Then I had to determine whether or not the extracellular matrix proteins that we were looking to stain for were present in the gelatin nanofiber itself, aka the scaffold before the seeding of the two primary cell types. And finally, I was to examine the expression of fibrosis-related ECM proteins in control and compare them to the TGF-beta treated 3D airway cultures. So the methodology began with the air-liquid interface, and essentially what that means is when you see the fibroblasts on the basal side of your 3D model, you put that in your growth medium. And then after about 2 weeks, you can seed your epithelial cells on top. Now, the epithelial cells are never exposed to the growth medium, but the fibroblasts kind of work together with the epithelial cells to keep them alive. And because they're always facing air, that's what allows them to differentiate into the ciliated cell type that you'd see in an actual human airway. Now, that does not occur in a normal petri dish model. And so that was completed before I was in the lab. Also, the treatment with the TGF-beta was completed before I was in the lab. So essentially, when I got there was a bunch of paraffin blocks of our 3D model, and they were blinded based on a number code system that Dr. Sam Wadsworth came up with. And so they were sectioned by our histology department, and I performed the immunohistochemistry stains on them. So then we took pictures with a light microscope, and we quantified the percentage of image per area of the stain that we happened to be testing for, and analyzed it statistically on PRISM. So at the bottom, I've included a table of our antibody targets, and I've put them in a pathological context as far as pulmonary fibrosis. So for instance, when we saw collagen 1 and collagen 3, we assumed this to mean that there was a stiffening of our fibrotic airway as long as they were present in fibers. And so how did we know if they were present in fibers? We also stained for decorin, which will decorate the collagen typically and organize it. Now, tenascin C is associated with detachment and thus mobility, and we'd expect to see this in cells that were attempting to repair themselves. Fibronectin is present in extracellular matrix during repair and fibrosis of airways. Thi1 is a fibroblast marker that will not mark myofibroblasts, while alpha-smooth muscle actin is specifically myofibroblast marker. Finally, Cas67 is simple cell proliferation. So this is the general results slide that I'm going to show you. In your top left-hand corner, we have the scaffold only, followed by the control that was not treated with any TGF-beta, and then a high-dose and a low-dose TGF-beta picture, which correlate to the graph, the low dose being the 1 microgram per milliliter dosage, and the high dose being the 10 micrograms per milliliter dosage. And so essentially what we saw from the collagen 1 stains, when you compare them to the control, is that we didn't really see a significant increase of collagen 1 as a result of the TGF-beta. However, collagen 3, we saw a significant increase between the high dose and the low dose when you compare them to the control. However, my collagen 3 low dose slides didn't actually turn out as well. As you can see, there's a little bit of destruction here on the picture, and we assumed this happened during antigen retrieval or the overall staining process. But moving on, decorin, in contrast to the collagen stain, seems much more localized, especially around the fibroblasts. And you can see this best in the high dose down here at the bottom. And we assume this to mean that this is where we'd find more organized collagen. And if we did have myofibroblasts in our 3D model, this would be the location on where they'd be pulling on these collagen fibers to stiffen the airway. And so, of course, we saw an increase when you compare the low and the high dose to the control, but not too much of a difference between the two dosages. Fibronectin, on the other hand, showed a somewhat blanket stain. And although we see an increase from the control to the low dose and to the high dose, this blanket stain, because it doesn't show any real specification throughout our 3D model, we assume that it was possibly being secreted in a somewhat soluble form. And that's why you see it kind of throughout the model. Next up, we have our alpha smooth muscle actin stain, which notably showed a high increase when you look at the high dose of TGF-beta. And so this was towards the bottom third of our 3D model. So this is what we assume to be our myofibrotic phenotype. And yeah, so you basically see it on the bottom third of our 3D model, but you don't really see much of it in the as a result of the low dose TGF-beta. So in conclusion for the simple single stain, we noted increased levels of collagen, specifically collagen 3, which would be associated with excessive matrix deposition, increased fibronectin, which is consistent with the fibrotic changes during the disease, and a possible myofibroblast phenotype. So these are all disease-relevant endpoints is what I'm trying to get at. And so the next experiment we decided to perform was the so-called serial sections. And that's where you take the exact same section of the exact same treatment group and you get like 5 cardinal sections of it, and then you expose them to your 5 different stains. So I was only able to complete this serial section methodology on the high-dose TGF-beta because that showed the most promise as showing results as far as like collagen 1 and collagen 3 increase. So when you compare collagen 1 and collagen 3 to Decker, and once again this is the exact same section of tissue, you see that although collagen 1 and collagen 3 seems to be present throughout the fibroblasts and gelatin region, the organized collagen is really only present on this kind of bottom third of our 3D model around cells that we were assuming to be myofibroblasts. So then when you compare the fibronectin stain to the alpha smooth muscle actin stain, we see in fact this is where, if we do have myofibroblasts, this is where they'd be located. They would have access to that organized collagen. They'd be able to pull on it and tighten our tissue. But also, these myofibroblasts aren't really associated with the fibronectin that's being secreted. And so that led Dr. Wadsworth to assume that either migrating fibroblasts that are taking residency in this gelatin region are the ones secreting the fibronectin, or the myofibroblasts are secreting the fibronectin in a soluble form to influence these migrating fibroblasts, and possibly a myofibrotic phenotype. And thanks to the specific air-liquid interface process of co-culturing, we do see differentiation, migration, and remodeling. And versus a Petri dish, this is a much more dynamic model. Now, the cons include that this is not an actual airway injury model. So normally for pulmonary fibrosis, something like asbestos or something like acid reflux will cause the actual injury to the human airway, which will then cause the release of endogenous TGF-beta. So we were currently working on a way to replicate this in our own model. And we also want to take more of a look at the variable effects of TGF-beta treatment on epithelial cell differentiation. For whatever reason, some of the 3D models, I didn't show any of them up there, but when they were exposed to the TGF-beta, the ciliated cell type actually differentiated backwards into a simple squamous epithelial cell type, and that you don't actually see in the pathology of the disease in humans. So we were trying to determine what that meant. But for our future directions, we would like to view our sections through multiphoton confocal microscopy. And this is specifically so we'd be able to see the 3-dimensional shape of collagen fibers, and that's kind of like a backup control to our Decker stain. We also wanted to perform Thigh1 and Ki67 optimization. So the Thigh1 optimization, you're going to change the pH of the buffer, and for Ki67 optimization, you're going to use biotinylated antibodies. If you'll notice, I didn't show any pictures of those two stains. I didn't actually get to optimize them in the time I spent at the Heart and Lung Innovation Center. And finally, we would like to, once again, come up with this epithelial damage-induced 3D model for our airway. And here's a list of my acknowledgments, and at this time, I would like to thank you all for listening, and we'll now open the floor to any questions, comments, or constructive criticism.
The downstream signaling of TGF-beta that are responsible for, I guess, the variable effects of treatment that you saw?
We weren't particularly sure. I mean, TGF-beta was just one of many of the profibrotic growth factors that we were going to test. So basically, when I got there, the overall 3D model construction, because we did use a 3D printer to make these, that was like just being finished, and I think TGF-beta was just the first growth factor that they happened to test, so we didn't really go too deep into like the downstream effects of TGF-beta, but that would have been a good question to ask my supervisor.
Related to that, do you know, because I'm not sure about endogenous concentrations of TGF-beta that happen after these micro-injuries and compared to what you're using 1 in 10?
Oh, okay. So I don't know the actual concentration of endogenous TGF-beta released. I just do know that that is part of the pulmonary fibrosis pathology seen in the literature, seen in like the disease pathology. So I couldn't give you an actual concentration comparison, and around this time now, he probably has like 3 or 4 different concentrations of TGF-beta, but I think he just wanted like a low dose to a high dose, and he never really told me why he chose the 10 micrograms versus the 1 microgram per milliliter.
So what are going to be some of your future directions when you say you're going to look at further epithelial damage, or what are some of the assays that you're going to?
So they wanted to initially introduce a mechanical injury. We tried using bleomycin initially. There's a whole half of my experiment I wasn't able to show. We tried to use the bleomycin poison on our 3D airway as a way of like a formal mechanical injury, but it eroded away all the epithelial cells. It caused like irregular fibrotic cells to be produced. So we're going to try and move away from a chemical irritant to more of a mechanical irritant, maybe some sort of a plastic scraper. That was actually what was used in the mouse model as a contrast to the bleomycin. They would just go in and physically injure it with a sort of a plastic scraper, and then they would kind of track the effects to see if you see an upregulation of TGF-beta or any of the other. But I mean, you don't see that in the mouse model just because the mouse model is, or the mouse epithelial cells are so different from the human airway epithelial cells.
So would it be harder to look at, to measure quantitate the damage if you use like a plastic scraper, or would it be another approach that you guys would use that will allow for you to better to quantitate the damage there?
I'm sure there's something better we could use than a plastic scraper. We probably will end up going back to a chemical irritant, like something probably similar to hydrochloric acid, because acid reflux was seen as one of like the predetermining factors to the disease. However, bleomycin itself was far too strong of a toxin and had way too many side effects, so we couldn't really separate out disease pathology to just the result of bleomycin specifically.
Thank you all for your work. I was wondering if you considered mucus secretion and clearance in this model, and if there's something to take into account which I tried to develop.
Yes, we did briefly consider the mucus secreting cells, and in fact in later models of the 3D model we are planning on introducing more cell types to show a more dynamic relationship. So the epithelial cells that we had didn't secrete, to my knowledge, as much mucus. It would have been like a significant disease-relevant endpoint to measure, but we did talk about incorporating specifically Clara cells and possibly a few other cell types to further show like a dynamic relationship between our 3D model and what you'd actually see in a true human airway. So that was one of the directions we were heading in.
Last question.
Once all determined what the best model is moving forward with replicating that damaged tissue,
Could you repeat the question from the beginning?
Sure, I said once fall is determined what a good model is to replicate those cells that you and cells, are you going to change that? You said you're not sure why you chose the one mechanism versus the one like you think you will be changing that once you figure out the best way to.
I'd imagine he'd change it to more closely represent, as I think someone else asked the question about how we selected those dosages. Was it based on endogenous levels of TGF-beta release? He'll probably try and make it like somewhat closer to that endogenous level. It probably won't be quite to that level, but he'll probably adjust the dosages down to, because I mean 10 micrograms per milliliter is probably way higher than anything you'd see in an actual human, even if they did have pulmonary fibrosis. But that's a good question.